This video is going to be on red blood cell indices. And the two red blood cell indices I'm going to talk about is the mean cell volume, or MCV, and the mean cell hemoglobin concentration, or MCHC. I'll first talk about causes for increases or decreases in the MCV. So the mean cell volume is the average volume of the red blood cells in circulation. So I've pictured here normal-sized red blood cells for comparison. If there's a decreased MCV, that means that on average, the red cells are smaller than the normal population of animals. And when we see this, we say there's a microcytosis or microcytic. The first cause of microcytosis is iron deficiency. So now remember that hemoglobin concentration is the stop signal for red cell division in the bone marrow. And iron is really important for hemoglobin synthesis. So if there's less iron, there's less hemoglobin synthesis, and then the red cells don't have that stop signal and they go on to have an extra division resulting in smaller cells. The second cause of microcytosis is liver failure or portosystemic shunts, or PSS. In both of these diseases, there's a decreased hepatic functional mass. Now, the liver is really important in making iron transport proteins. So the iron needs to get to the bone marrow to be used for erythropoiesis. If there's less iron transport proteins, there's less iron getting to where it needs to go. Again, there's less hemoglobin in the red cells, and then another division during erythropoiesis resulting in smaller cells. The next two causes of microcytosis are normal variations. First, some Asian breeds like Akitas and Shibas normally have smaller red cells than other breeds. And young animals, less than a year of age, tend to have smaller red cells compared to adults. So if you're looking at a young animal MCV with an adult reference interval, it might be lower. Okay, so now I'll go over to larger red cells, or an increased MCV, which is known as macrocytosis. The first and most common cause of macrocytosis is red blood cell hyperplasia or regeneration, where there's younger red blood cells, reticulocytes and polychromatophils, in circulation. These cells are larger, so they increase the MCV if there's enough of them in circulation. The second cause of an increased MCV is FELV infection in cats. With FELV infection, it's a form of dysplasia. The red cells actually skip a division, and that results in larger cells. Third, normal breed variation in some dogs. Miniature poodles, toy poodles particularly, have larger red blood cells. And then the last cause of macrocytosis is an artifact, either with agglutination or storage. With red cell agglutination, there's antibodies holding red blood cells together, and they're forming doublets and triplets of cells, but the analyzer doesn't recognize the doublet and the triplet. It just sees it as one big cell, not two normal-sized cells. So it sees a larger and only one cell. Okay, so now I'm going to go down to MCHC. When there is less hemoglobin per red blood cell, that results in a decreased MCHC, which we call hypochromasia or hypochromic. We see this most commonly with red blood cell hyperplasia or regeneration, and that's because those younger red cells, the polychromatophils and the reticulocytes, they have less hemoglobin content per cell, because they're actually still making hemoglobin when they're that young. So they just haven't made as much hemoglobin as they're going to in their adult or mature forms. The next cause is iron deficiency. So if there's less hemoglobin per cell, if there's less iron available for hemoglobin synthesis, then there's less hemoglobin being made, and that results in less hemoglobin per red blood cell. So now going over to an increased MCHC, and this actually is not physiologically possible. So if you see an increased MCHC, that has to be an artifact. And common causes of this artifact include hemolysis, Heinz bodies, and lipemia.